Today, a delay on a regulatory decision in lung cancer, an orphan drug status in breast cancer, disappointing findings in glioblastoma multiforme, a European approval in renal cell carcinoma, and a Chinese approval in lung cancer. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has extended the review period for a supplemental biologics license application for atezolizumab for use in combination with carboplatin and nab-paclitaxel as a first-line treatment for patients with metastatic, non-squamous, non-small cell lung cancer who do not have EGFR or ALK aberrations. The extension will allow ample time for the FDA to review additional information it requested for the application. The new action date for the sBLA is December 2, 2019. In January 2019, Roche, the manufacturer of atezolizumab, reported that the FDA had granted a priority review to the sBLA based on findings from the Phase 3 Empower 130 trial. Results of the study demonstrated a statistically significant improvement in both progression-free and overall survival with the atezolizumab triplet compared with chemotherapy alone in patients with stage 4 non-squamous NSCLC. Data from the interim analysis showed that the atezolizumab arm was superior in OS in the intent-to-treat wild-type population. The median OS was 18.6 months versus 13.9 months with carboplatin and nab-paclitaxel alone. Moreover, the 1- and 2-year OS rates with atezolizumab were 63.1% and 39.6% versus 55.5% and 30.0% in the carboplatin nab-paclitaxel arm. In breast cancer, the FDA has granted an orphan drug designation to neratinib for the treatment of patients who have brain metastases. Prior data have demonstrated the activity of the small molecule TKI in combination with capecitabine in patients with HER2-positive breast cancer who also have brain metastases. For example, results of the Phase 3 NALA study showed that fewer patients required intervention for central nervous system metastases with neratinib and capecitabine versus lapatinib and capecitabine. At 54 months, the cumulative incidence for intervention for CNS metastases was 22.8% versus 29.2% for the neratinib and capecitabine arms, respectively. Moreover, additional study cohorts from the Phase 2 TBCRC-022 trial showcased promising results with a combination of neratinib and capecitabine in patients with HER2-positive breast cancer and CNS metastases. Neratinib is currently approved by the FDA for the extended adjuvant treatment of adult patients with early-stage HER2 overexpressed amplified breast cancer following adjuvant trastuzumab-based therapy. The combination of nivolumab plus standard temozolomide and radiation therapy did not show a statistically significant improvement in progression-free survival versus temozolomide and radiation therapy alone in patients with newly diagnosed glioblastoma multiforme that is MGMT methylated, missing one of the primary endpoints of the Phase 3 CheckMate 548 trial. A data monitoring committee did recommend, however, that the trial should continue as planned to allow overall survival, the other primary endpoint, to mature. Bristol Myers Squibb, the developer of the PD-1 inhibitor nivolumab, stated in a press release that it remains blinded to all study data. Disappointing top-line findings for the Phase 3 CheckMate 498 trial were previously announced in May 2019, which evaluated nivolumab plus radiation therapy versus temozolomide in patients with MGMT unmethylated GBM. In this multi-center study, results showed that the nivolumab regimen did not improve OS over temozolomide, missing the co-primary endpoint. In renal cell carcinoma, the European Commission has approved the combination of pembrolizumab and axitinib for the frontline treatment of patients with advanced disease. The approval is based on findings from the Phase 3 Keynote 426 trial, in which the combination led to a 47% reduction in the risk of death versus sunitinib in this patient population. The pembrolizumab and axitinib combination also showed an improvement in both progression-free survival and objective response rate versus sunitinib. Additionally, at a median follow-up of 12.8 months, the median OS was not reached in either arm. The median PFS was 15.1 months for pembrolizumab and axitinib and 11.1 months with sunitinib. With the combination, there was also a 31% reduction in the risk of disease progression. With the approval, the combination is now available for use in all 28 European Union member states, as well as Iceland, Liechtenstein, and Norway. The regimen was also approved by the FDA in April 2019. In non-small cell lung cancer, China's National Medical Products Administration has approved osimertinib for the frontline treatment of adult patients with locally advanced or metastatic non-small cell lung cancer whose tumors harbor EGFR exon 19 deletions or exon 20 L858R substitutions. The approval is based on data from the Phase 3 FLORA trial, in which frontline osimertinib reduced the risk of progression or death by 54% versus standard TKI therapy with erlotinib or gefitinib. In the double-blind study, the median progression-free survival was 10.2 months for standard therapy and 18.9 months with osimertinib. Additionally, the PFS benefit with osimertinib extended across all pre-specified subgroups. The objective response rate with osimertinib was 77%, compared with 69% for erlotinib and gefitinib. The median duration of response with osimertinib was 17.6 months versus 9.6 months in the comparator arm. Osimertinib was previously approved in the United States and Europe for the frontline treatment of patients with EGFR-positive NSCLC. This week, we sat down with Dr. Hussein Taubi of the University of Texas MD Anderson Cancer Center to discuss BRAF and MEK in melanoma.
So in terms of what the role of BRAF mutation is in melanoma, actually up to half of patients with melanoma have a BRAF mutation in their tumor. And in those patients, the BRAF mutation actually is oncogenic, which means that it actually drives the development of the cancer. We actually understand very well kind of the pathway that's involved in that. It's called the MAP kinase pathway. And it's basically, because of the mutation, we have aberrant signaling through that pathway that drives the cell into continuous proliferation and growth. And actually now we also know it affects the tumor microenvironment to make it less immunogenic as well and less responsive to immune therapy. And so those mutations, again, appear in about half of the patients. Very specifically, there's about 80 to 85% of those mutations are V600E. Another 5 to 7% are V600K, and then there's a smaller percentage of other mutations that could be relevant. So the presence of a BRAF mutation, from my perspective, simply gives us additional alternatives for those patients in terms of using targeted therapy. In the past, there used to be some kind of questions about the prognostic significance of having a BRAF mutation, where it seemed to be a bit more aggressive disease. But again, I think the landscape has changed because now we have therapy that can alter the outcomes of those patients. So the BRAF mutation is a mutation that is that affects the BRAF gene, which is actually part of the MAP kinase pathway. And the MAP kinase pathway is basically a signaling pathway that starts at the cell surface, signals through RAS, then RAF, then MEK. And ERK is the last protein that actually gets into the nucleus and changes the expression of genes and so on. So that kind of pathway is sequential. And when we have a BRAF mutation, the BRAF itself becomes the oncogenic signal, so to speak, and just kind of takes over and starts firing through that pathway and activates MEK, activates ERK, and leads to cell proliferation. So the first targeted therapy that was very specific to the mutated BRAF protein was actually vemurafenib. And now we have dabrafenib and encorafenib, and those three drugs all go to the mutated BRAF gene, blocks the signaling through it, and by doing that shuts down the oncogenic signaling through the through the MAP kinase pathway and leads to cell death, and also, as I discussed a little earlier, actually changes the tumor microenvironment to a more favorable for immunotherapy as well. Now, the interesting part is when you inhibit the BRAF gene alone with the BRAF inhibitors, that's effective, and that shuts down the oncogenic signaling through the MAP kinase pathway. But the cell actually the cancer cell tries to figure out ways to bypass it. And so the vast majority of the time, about 50% actually through reactivation of the MEK, and another 25% also involves activation of MEK. So I usually summarize it as 3/4 of the time. You basically have multiple different mechanisms that all kind of hone on MEK and reactivate signaling through MEK itself. So that actually made it a really easy target because MEK inhibitors were in development as well. So we have trametinib, we have binimetinib, and we have cobimetinib, and those are three MEK inhibitors that are capable of, again, shutting down the signaling through that pathway through MEK. And so as we started dealing with BRAF inhibitor resistance, and we figured out that, again, 3/4 of it was driven through MEK, it made a lot of sense to combine BRAF and MEK inhibitors. And obviously that ended up being more effective, quite significantly more effective. We increased response rates, we increased progression-free survivals, and interestingly enough, it ended up being less toxic in general. It improved the toxicity of BRAF inhibitors, also, although it kind of generated a few new toxicities that were not as significant before, but improved patient outcomes. And at this point, we almost never use BRAF inhibitors alone. We always use the combination of a BRAF and a MEK inhibitor.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.